Mutation is an alteration of the nucleotide sequence. Mutagens are what cause a mutation, and they can be in the form of a chemical or UV change. Spontaneous mutations are mutations that occur without a mutagen. Mutations can also occur from errors during DNA replication, mitosis, meiosis, or even damage to DNA.
There are many types of mutations. There are 3 types of base pair substitutions. The first is a silent mutation, which is a change in a base pair, but it still codes for the same amino acid. In this original DNA strand, you can see that this base pair codes for Phi. In the mutated DNA strand, the mutation changes from an A to a G, which still codes for the amino acid Phi. The second type of base pair substitution is a missense mutation, which is when a base pair changes as well as the amino acid. You can see that this base pair codes for the amino acid Val, but in the mutated DNA strand, it changes from a C to a G, which then codes for the amino acid Lu. The third type of base pair substitution is a nonsense mutation, which is when there is a premature stop codon. In the original DNA strand, you can see that the CTT codes for the amino acid GLU, but in the mutated DNA strand, it changes to ATT, which then codes for a stop codon before the original stop codon. There are also 2 types of frame shift mutations. The first one is a deletion mutation, which deletes a base pair and changes all of the amino acids. In the original strand, you can see that there is a T in the CTC base pairs, which codes for the amino acid GLU, but in the mutated DNA strand, the T gets deleted, which then makes the mRNA GGG instead of GAG, which then codes for the amino acid GLY, which changes all of the amino acids after that, as you can see. The second type of frameshift mutation is an insertion. An insertion is when a base pair is inserted into the sequence and it changes all of the amino acids. In the original DNA strand, it is CAG, CCC, ACT, which codes for the amino acids GLN, PRO, and THR. In the mutated DNA strand, it is then CAG, CT, which gets inserted, CC, ACT, which codes for the amino acids GLN, GLU, and VAL, which is different from the original DNA strand.
So the first type of repair is called a base excision repair, and this is when a damaged nucleotide is repaired or replaced. So look at there. Oh my gosh, it's broken. We're recognizing it's broken. So we're going to remove the broken piece and boom, fix it back together normal. The next repair we have is the nucleotide excision repair. So this is when a short stretch of damaged DNA is replaced. So instead of just one chunk like the base excision pair, we're looking, oh my gosh, damaged. We need to delete it. So we separate them and then boom, boom, boom. Here's the newly replaced segment. And the last type of repair is the double strand break repair. So this is a type of recombination, and it is a repair done by ironing the radiation, and you need a second chromosome for this to be complete. So as we can see, they're doubly broken. We're gonna separate them, and then we fix them at the end.
As you can see, some mutations impact DNA strands and some do not. Some alter the amino acid product or prevent a gene from functioning properly or completely. Missense mutations can cause sickle cell disease and epidermolysis bullosa and SOD1-mediated ALS. Nonsense mutations account for 11% of human inherited disease and 20% of disease-associated single base pair substitutions. Overall, a mutation is an alteration of the nucleotide sequence, and it can occur from many things.